So we're very excited to kick off our series in CART with our esteemed colleague, Helen Tager-Flusberg, who's from Boston University. So I looked at her CV. I've actually never even seen her CV before. Holy crap. So 7 books, 7, wow. More than 200 empirical articles, 61 chapters. I hate writing chapters.
Me too.
61? Wow. So she's obviously a very esteemed scholar. But her work is really in the area of the development of language in kids with autism, but also individuals with other neurodevelopmental disorders. And she started off very early in her career looking at characteristics of kids with ASD and how that might compare to other groups of individuals, and really theory of mind as well as it relates to language. But now she's kind of moved further afield and is actually combining behavioral measures, in fact some language measures that she's developing, along with biology. So EEG, fMRI, and she's studying more with a focus on infants at risk for autism and also minimally verbal children with autism. So she's kind of a pioneer in that area of minimally verbal. She had an ACE center and now has a P50 center grant with UCLA. So she's involving us in her work, which is really great. And the best part is that she's going to be in residence at UCLA January to March next year. Mostly at UCLA or maybe mostly at Venice Beach, we're not sure. But she'll be here. And so we're really excited that Helen's come to talk to us today about language and autism.
Thanks so much, Connie, for that lovely introduction. And thanks to all of you for inviting me. It is really just such a pleasure to be here today. Seeing how things are here, getting used to wonderful Los Angeles weather, and really to see what a very enriched environment you have here. So I titled my talk Explorations in Language because I sort of wanted to kind of give a broad scope. I'm going to start by talking about the place of language in autism, talk about language impairment with familial patterns seen in other family members, and some clues to neural biomarkers. Then I'll move into the study of high-risk infants, looking at early development and neural predictors, and then speak a little bit about the work that we've done already, which is a kind of a background on the minimally verbal that led to this wonderful new so-called clinical research center program that we have now in collaboration with you, and draw some conclusions. Okay, I'm going to jump right in and talk about how I view and everyone else views language. So there's really 2 different components. First, it's a code for transmitting information. Call that part the linguistic code. It involves the sounds, the semantics, syntax, etc. But it's a code for something. What is the function? And the function is for effective communication. The linguistic code serves the function of communication along with non-linguistic cues in a range of social contexts. Okay. When we think about ASD as it's defined today, we have, of course, our 2 core domains of social communication impairments and restricted repetitive behaviors, but they're surrounded by a whole bunch of co-occurring conditions. Okay. And most importantly, language impairment itself is now considered a key co-occurring condition. It's no longer a core-defining feature. So when we think about language and communication in autism, there are, of course, core universal impairments in the social code that defines language, and in essence, that is social communication. But then there are these co-occurring comorbid impairments in the linguistic code. And here we see amazing heterogeneity in outcome, from the nonverbal, people who never acquire a single spoken word, to those who have superior linguistic skills, including one young man I know who so far has mastered 9 languages. He never quite gets through because he can't get to the literature parts that you need to do in college, but he just loves language as a linguistic code. And of course, there's everything in between. Why is it still important to study linguistic ability if you think you're an autism researcher? Well, it is, as we know, the single best predictor of long-term outcomes, over and above the prediction that we can make from intellectual functioning. So when we look at the landscape, this is just a cartoon, really, of the different groups that we can identify, from the minimally verbal to those who have no language impairment, or what I call the ALNs, about 25%, and the verbal but language impaired, which make up probably almost half the population. And for the most part, most of my career and most of the literature on language and autism focuses on the kids who do acquire some level of verbal functioning. And what we've been interested in for the last 20 years or so is to understand this kind of heterogeneity. And we began by looking at the heterogeneity and the different language profiles that with my postdoc at the time, Margaret Kjelgaard, in 2 groups. This was a large cohort, our initial CPEA study, for those of you who remember back to those days. And we found that there were 2 groups of kids based on performance on standardized language tests. Indeed, the ALN, who you can see have a flat profile with an average score of around 100, which is average. And then this ALI group, autism but language impaired, who these were older kids. They did not have articulation problems, but they were impaired in non-word repetition, vocabulary, as well as higher order syntax and semantics. And this was an interesting profile, which we and others drew attention to the similarities to the profile that you see in kids with what's now known as developmental language disorder. We then went on to look at family studies. We wanted to know to what extent do these language impairments travel in families of kids with autism. And my then doctoral student, Kristen Lindgren, focused on this. And we found that we were looking at the unaffected siblings and the parents, okay? The unaffected sibling had lower language and reading scores if the autistic sibling had language impairment rather than normal language. And here you can see the scores in language on the left and reading on the right, divided on the basis of whether their sibling had normal or impaired language. And then parents and siblings of children showed increased rates of history of language disorder, whether it was developmental language disorder or dyslexia. And we weren't the first to show this. When we looked at parents, for the ALN group, you can see here it's the fathers. Oh, the colors are wrong. The fathers who have about a 20% history, mothers very low. But when you looked at the ALI group, the language impaired, parents of the impaired kids, it's both have significantly higher rates, but the difference in the mothers, that is quite striking here. So it's clear that language problems are traveling in families that also have a child with autism spectrum disorder. You know, there's a couple of places up front for people to sit. Okay, so then we began to look at the brain, okay, and we know that in language, in the brain, in typical individuals, language, particularly linguistic code aspects of language, are more represented in the left hemisphere, in both this frontal Broca's area and in the temporal parietal region. And here I highlight the planum temporale, which is part of Wernicke's area, and the area that we looked at in our initial structural imaging studies. Okay, and this is just to show that we, of course, did a lot of work preparing our children. It's not quite as easy as it looks here, and that's my granddaughter at the Philadelphia Science Museum. Okay, so one of the things we looked at, again, my student Kristen Lindgren looked at the asymmetry of language areas in autism, children with autism, controlled children, and the siblings. And the siblings here are represented in green. And you can see that we see exaggerated left hemisphere asymmetry in the autism group compared to controls. And the siblings are showing an intermediate pattern here. They're not showing the reduced asymmetry in the frontal regions. However, here they're traveling more with the typical controls. And then we turned our attention to fMRI studies, and the very first study was done by Marcel Just, and then we followed on with a study of semantic processing in these initial studies. This is kind of ancient history now, but it's actually interesting. It was just 15 years ago, the first fMRI studies. And in those studies, we started out with adults with autism and found reduced activation in the frontal region, which we saw is structurally smaller in the left hemisphere. But importantly, these studies highlighted reduced statistical connectivity between the frontal and the temporal activation sites. And those are that connectivity is key to processing language. And then with my, at the time, postdoc Tracy Knaus, we turned to adolescents, and instead of doing a receptive task, we did a generative language task. Again, we found less left hemisphere asymmetry in activation, and again, reduced correlation between the frontal and the temporal activation. So to summarize this kind of background, or the first, 10, 15 years, we see that there's a heterogeneity in language profiles, as well as in family history, and it's reflected in that. We see atypical structural and functional asymmetry in the cortical language regions, which is an endophenotype present in siblings. Reduced functional connectivity between the frontal and the posterior regions we've also found, but that had not been looked at in siblings to date. So, more recently, we've turned to, as Connie pointed out, became very interested in looking at the very earliest developmental stages, thinking about the developmental origins. And of course, our work is not just about predicting language outcomes, but like everybody else, particularly the wonderful group here, looking at trying to predict autism outcomes. But I'm today going to be focusing on the language outcomes. And my own interest in this is to identify risk factors for language impairment very early in development. And we do so by studying high-risk infants, those with an older sibling. We study them longitudinally from soon after birth. We've begun our studies at the age of 3 months. We've now extended this work to look at other high-risk groups, but we're not able to do so in the first year of life, or at least with the cohorts we've been studying. And everything I'm talking about here is a collaboration with Professor Charles Nelson from Boston Children's Hospital. And in our work, we're trying to pack everything in because he and I bring very complementary and distinct areas of expertise. So we're looking not just at language, my expertise, but also faces and social, emotional functioning, which is his. And we have all kinds of brain and behavioral measures. And this gives you a snapshot of that initial cohort of studies in which we managed to enroll almost 260 children. And we do electrophysiology, some eye tracking, behavioral measures, and other measures. And the ENIRS goes down here because this part was optional and kind of under separate funding. And in red are the examples of the data that I'm going to talk about this morning. So let me go back and ask the question of high-risk families, okay? To try and put in context, we were really interested, we came along as in the second wave of researchers investigating high-risk infants, and we really wanted to know, at least my doctoral student, Megan Talbot, was really interested in what's the context. Think about these families who we are enrolling in our study. And they've already got a child diagnosed with autism. Fully 1/4 of our sample of the high risk for autism group did not discover that their older child had autism until they were pregnant with their second child. So that's an interesting context to put this in. So what's it like to raise a child? And we've done lots of work around this, but I'm just going to give you a small snapshot from the weekly diaries that parents filled out, both in our high-risk group as well as in our low-risk group, between the ages of 6 and 12 months, because we wanted to know prospectively what are parents worrying about, what kinds of signs might distinguish between our groups of infants very early on before they know anything about, they or us know anything about outcomes. And embedded in a set of kind of nice, fun questions, and what did you do this week, and any new things in your baby, we asked if they had a concern about their baby, and if so, what that concern was. Well, you can imagine, or anyone in the audience who's ever had a child, everyone has concerns about their baby. I can tell you that then we coded the kinds of concerns that they raised. We think about, the interest in the autism world in sleep and medical, gastrointestinal. Yes, guess what? All mums are worried about diarrhea this week. Lots of babies have sleep problems. These do not distinguish at all between high and low risk babies or in infants who go on to develop autism. None of the medical side stuff did. Behaviorally, we then coded what they talked about in terms of whether they were talking about social communication problems, repetitive behavior, sensory issues, and language. And I'm just going to give you a snapshot of the concerns raised at the age of 12 months. And in red, you can see the infants who were later diagnosed with autism. Green, the high-risk infants who did not end up with autism, and the low-risk controls. And actually, by 12 months, control moms are not very worried anymore. The high-risk moms who don't end up with autism still have lots of worries, but we see that it's language that is really the distinguishing concern, and this kind of reflects prospectively exactly what we've read in the literature going back many decades now, that what parents bring as their initial concern to pediatricians is they're worried about their children's language. So what are they actually worried about? Well, my then doctoral student, Karen Chinowski, looked at the infant's vocalization rate in the lab at 12 months, and we found that indeed the infants later diagnosed with autism vocalized less. Okay, we're not talking about words or sentences. This is 12 months speech vocalizations. So they're certainly vocalizing less. And then when we look at the developmental trajectories for expressive and receptive language on the Mullen, we see in these developmental trajectories that the group later diagnosed with autism begins to diverge at around 12 months. And actually, even at the beginning, they are lower. Here, the green and blue colors are reversed. But we certainly see that both the siblings and the infants later diagnosed with autism have lower language scores. But I want to point out something really important to keep in mind, because I'm going to be talking about risk factors for language. But all our kids acquired spoken language, and in fact, for the most part, their language was really pretty good for their age. And we can talk in the question area as to why that was. There's probably several reasons. But I want to point out this is still a relatively high language group, but nevertheless, there's still a lot of variability within these groups, and there's also overall lower. So why are the high-risk infants, even those not diagnosed with autism, falling behind in language? Because the differences grow, especially between the ages of 24 and 36 months. So my former student, Mayor Chita Tegmark, looked at this in an eye tracking paradigm. So we looked at eye tracking measures in the toddlers to measure the processing of single words. So they would hear, they'd be sitting in front of the eye tracking monitor, and they'd hear, "Look at the dog." And they'd see two pictures, the dog and the bird. And actually the pictures were up before they hear the phrase. And as you can see, the green dots representing eye fixations. You look more at the dog if you understand that word. But we're able to measure the reaction time, especially if the infant before they hear, "Look at the dog," is looking at the wrong picture. How quickly were they able to switch to the target? Okay, we did this at 18, 24, and 36 months. And we eliminated the kids who ended up with an autism diagnosis. We were really interested in these siblings in the endophenotype. And here are the data. On the left are the low-risk infants. We see the reaction time in blue for 18 months, purple for 24, and gold for 36. And on the left are the low risk, and on the right are the high risk. And the main difference is at 36 months. We see that the low-risk control infants show significantly more rapid processing, more efficient processing by 36 months, and they're able to sustain their fixation on the correct target longer. And we don't see that difference in that change. So at 24 and 36 months, the high-risk children are looking the same. So they're showing a kind of a less mature, efficient speech sound processing at 24 to 36 months. And we then turn to, so what are the underlying brain mechanisms? What might explain why they are less efficient language processes? So I'm first going to talk about our EEG data, where we are collecting baseline, or sometimes called resting EEG. And this is how we do it. It's 5 minutes of continuous EEG recording. And Chuck Nelson spends his time in the lab blowing bubbles to babies. Okay. And we've been collecting these data for on the infants at various ages. Now, our very earliest study, we still didn't have 3-month-olds, and we still didn't have 36-month outcomes. So what I'm showing you here are the developmental trajectories of power, the different power bands, power frequency bands from delta, theta, doesn't really matter, low, high, alpha. Basically what you see here is that the solid line represents the low-risk infants, and the dotted line represents the high-risk infants, which is a mix between those who did or did not end up with autism, because we didn't have the outcomes yet. And this was work that was done by Adrienne Tierney when she was a doctoral student. And we can see that in every power band, we see that The infants who are at high risk have lower power, although by the time they're reaching 24 to 36 months, they're approaching the low-risk control infants. But it's that difference in developmental trajectory that is most striking. Okay, and we've replicated this in different ways. More recently, Carol Wilkinson, who's a clinical, I don't know her title, she's a wonderful developmental pediatrician with an interest in language in Chuck's group, showed that reduced frontal gamma at 24 months predicted better language in the high-risk infants. So here what I'm showing you on the left is the data for the low-risk controls, the high-risk who don't end up with autism, and then the ASD group. And you can see that this sibling group, if you like, siblings who don't end up with autism, have lower frontal gamma at 24 months. Intriguingly, only in the high-risk group, lower gamma in those frontal regions predicted better language scores as measured on the Mullen. And at this point, we have high hand-waving possibilities to explain this. This was not an expected finding, and so we don't really have a clear sense, but this is something clearly that we should be pursuing in future studies. We would have expected perhaps higher gamma to predict better language. Okay, so that's the work from the just resting baseline EEG, okay, the general functioning of the brain. We then moved to electrophysiological measures as the infants are now processing speech. And we were interested here in looking at whether, I already talked about the atypical brain asymmetry in children and adults with ASD, including their siblings. And we wanted to know whether when high-risk infants would show atypical lateralization, and then also the reduced functional connectivity that we saw in our fMRI studies with adolescents and adults. Do we see that same reduced functional connectivity between frontal and posterior language regions as they're processing speech? So we have very simple paradigm. The babies are, again, sitting on mom's lap, and they can be watching a silent video or bubbles, and they hear just a sequence of sounds, das and tas and Hindi das thrown in there. And I'm just going to focus on the late slow wave that we measured at 3, 6, and 9, 12 months, because this is considered a measure of lateralization, sort of at the point sort of after about 300 milliseconds of listening to the sound, you see, in adults certainly, an advantage to a slightly faster processing in the left hemisphere, and EEG is very well suited to capturing those differences. So on the left are the low-risk controls, on the right are the high-risk who did not end up with autism. This was the work of my doctoral student, Annie Siri. And at 6 months, which is this top row, okay, this has died, we see there's no differences in the two lines which represent the left and the right hemisphere between either the left, the low risk or the high risk infants on the right. By 9 months in the middle group, we see that the low risk controls are showing a significant difference in the processing with the advantage to the left hemisphere, but not in the high risk infants. And again, the same at 12 months. So we don't see differences in the high-risk infants at any age in lateralization. So this atypical pattern of lateralization seems to start quite early. And then in a follow-up using a larger sample where we had a larger group of autism outcomes, my student Kayla Finch looked at this and found that these are the averaged waves at 12 months. In the top band, we see the low-risk controls who are showing the left hemisphere advantage, no lateralization in the high risk, no autism, but a reversed lateralization, which is depicted here in the infants who end up with autism. So again, it sort of reflects that sibling group who don't end up with autism are somewhat intermediate and are showing this as an end of phenotype. And then we looked at functional connectivity. So in this same paradigm, instead of looking at the event-related potentials, we looked at gamma coherence and used a measure of intrahemispheric. So looking at the connectivity within each hemisphere, linear coherence in the gamma band at 6 and 12 months. And this was Julia Righi and Adrian Tierney's work. And what we found was reduced functional connectivity, not at 6 months, but certainly by 12 months in the high-risk infants. And you can see there's increased connectivity, or increase in the measure that taps into connectivity in the low-risk infants, but not in the high-risk infants. And then when we separate out those infants who ended up with a diagnosis of autism, again we see that familiar pattern with the lowest connectivity in the infants who end up with autism, and the siblings without autism are intermediate. We did a similar connectivity analysis using, there's overlapping infants for this, but a very different methodology and language paradigm. So here we were using functional near-infrared spectroscopy, which uses light to detect changes in oxygenation levels in the brain. And it's quite well suited for language studies because it detects blood oxygenation levels at the surface, and the language regions are there. So we've become quite enamored of using ENIRS. So in this, we used a different paradigm. It's a kind of a familiar statistical learning paradigm. We gave syllables. The babies heard different syllables, 28 blocks of artificial words, repeated either patterned as in penana or ABC kinds of patterns like baloti. So they're just listening to this, just goes on and on and on, and we're measuring their blood oxygenation levels. And Brandon Keene did this work, and here we were able to include our 3-month-old infants, which turned out to be extremely interesting. And here's just another cute picture. This is what the cap that we use to put the probes to detect, to emit and then detect the emitters and the detectors of light on several different cute babies. And you strategically place the probes depending on your task. And here we're looking at both left frontal and left parietal brain regions as depicted here. And so in our regional connectivity analysis, which is all I'm going to present to you here, we're looking at how connected, how similar in timing is the change in oxygenation level in these different regions of interest in both the left and the right language areas. And what you see here is this is on the left 3, 6, 9, and 12 months. The colder the color, the more blue, means greater connectivity in the high-risk infants. Okay, the warmer or the redder the color, the greater the connectivity at 12 months. And what you see here is that at 6 and 9 months, we have no group differences. But at 3 months, in fact, there's greater functional connectivity in this language task in the high-risk infants. By 12 months, we see the reverse. So these data with fNIRS completely replicates what we saw with the EEG data at 6 and 12 months. But by adding in 3 months, we see, now wait a minute, they don't start out lower. They're actually starting out with higher connectivity, and there are very significant changes happening early on. And unfortunately, too few of us are studying these very young infants, and there's obviously lots of very interesting differences there. Unfortunately, in our current study, we're also not able to follow up on such young babies for the most part. Okay, for the final part of my talk, I want to turn to the minimally verbal kids. Okay, just to give some background and to sort of fill out the picture, because really everything I've talked about so far is about developmental patterns and risk factors and biomarkers for language in kids who do acquire spoken language. But then we have this 30% or so who never do acquire much, if any, spoken language. And what do we know about them? And the answer is we know very little. But for the last 5 to 7 years, we've begun to investigate this. And what we focused on, and this was a big collaborative ACE research program, was on why don't they speak? Okay, and I think we've got a few clues to this at this point, but I would say we still, are just beginning to scrape the surface. And one of the huge challenges, and the reason that no one until fairly recently has really ventured into doing much work with this population, is that they're extremely challenging. The measures that we would use to assess their behavior are not necessarily valid. And actually, Connie, Kathy, and I chaired a work group and spent a year trying to figure out what would be the best standardized behavioral measures. And we came up with a list that I don't know makes us enormously proud, but it was definitely the best that there is. So I just want to put that as background. But truly, for someone who has spent her whole career focusing on language in this population, I had reached a point where I felt, really, Helen, you are just missing the most important question in the field, which is the ones who don't speak. Studying those who do is interesting, has given us, has been very enriching, but really this is where we need to focus. So that's what we've been doing. Now, why don't they speak? Well, there's lots of potential explanations and more beyond this, but I've mentioned these here because this is partly what we focused on. They don't have the motivation, or they don't understand intentional communication, and maybe that's indexed by joint attention, symbolic deficits exemplified perhaps in play, impaired imitation, intellectual disability, speech motor impairments, sound processing, and other neurobiological mechanisms which may be more profoundly impaired, particularly those language regions critical for language processing. And the answer is there's no correct answer here. They're all right. Okay. We started out, my student Meredith Pekukonis took a bunch of, we had done some pretty rich behavioral phenotyping, and we had lots of measures on, this is a group of 43 children and adolescents, all of whom were minimally verbal, defined as they couldn't get beyond a mod 1 on the ADOS. And we had measures of joint attention, including behavioral regulation, initiation, response. We had a play measure. We had imitation measure, measures of cognition or IQ, nonverbal IQ. And of course, they were widely varying in age, so we thought age may be somewhat of a predictor. Because within this group, there's still some variation from no language to no speech to some, but just simple phrases and sporadic or maybe just echolalic. So when we looked at these correlates, basically what we found was that the only three domains that did correlate in this much older group were play, imitation, and IQ. And then when we controlled for IQ, play and imitation were still significant predictors, even beyond IQ differences. And then in a regression analysis, it was really imitation. And this is not oral imitation, this is motor imitation, which of course conflates motor measures, imitation capacity, if you think about what any imitation test involves, there's a lot packed in there. So we don't quite know, that may be why it turns out to be the most significant predictor, but it does highlight that for us. Then my former graduate student, now postdoc, Karen Chinowski, who's always been a speech expert and spent many years trying to persuade me that looking at speech rather than language was what's really interesting in autism. I remained skeptical and patronizing for quite a few years, and then have become now, I have to tell you, a true believer. And one of these days will in fact go back to school and take courses in speech, learn how to do phonetic transcriptions, and learn much more about speech motor functioning. So she had the idea that maybe in listening and observing these kids, that many of these minimally verbal looked like they had childhood apraxia of speech, which is a rare disorder, and it's about the precision and consistency of speech output, but it's not peripheral, it's brain. Okay, and so asking the question whether minimally verbal, some of them at least, have co-occurring CAS. Now, CAS, there's lots of controversy about how do you define it. At the end of the day, the child has to produce enough speech to be able to code the kinds of signs of CAS. If you don't produce distorted speech, if you don't produce speech, you can't tell whether it's distorted or not. And we did this work with two colleagues from Australia, Amanda Bricknell in the middle and Angela Morgan, and they're from Melbourne. And they co-coded, so reliability, because not a lot of people have the expertise. And in this country especially, there's a little bit of a CAS mafia, and it becomes somewhat, which we can talk about in private meetings. Okay, so signs of CAS, there's when you're trying to produce a word, and this is how we measured it. They were given a word that they were supposed to be trying to copy, okay? And it's not, you actually give them the model, the speech model itself. Voicing problems, consonants, vowel errors, all kinds of, there's just a distortion in the speech itself. And what we found indeed was that there were high rates of CAS among the minimally verbal. And I realize this is probably not so clear to read here. These were the numbers of children. We had 57 kids. There were about 12 of the 57 who had no impairment at all. So speech motor CAS problems are not, that's not the only explanation, okay? So these kids didn't acquire spoken language, but they don't have a problem. A larger number had a speech disorder, about 16 of them, but didn't quite meet criteria for CAS. At CAS, we coded in about 25% of this group, but fully 1/3 of another 25% didn't produce enough speech. And you have to ask, is that an even more profound variant of this? So over 75% had some speech motor disorder. And interestingly, we looked at their vocabulary, the number of different words that they use in a spontaneous speech sample, and about a third of the variance in how many different words they knew is actually accounted for by the speech motor abilities. So there's a much closer connection between speech motor functioning and the acquisition of vocabulary in this population, as it is actually in typical kids, but we tend to sort of forget those connections when we look at this. I'm going to quickly go through this. We looked at auditory processing, and this was with my colleague Barbara Shin-Cunningham and Lawang, a postdoc. And this is a typical mismatch negativity paradigm. They hear complex tones, and it's either a simple oddball. We're just looking at how the brain detects. The kids got to do nothing. This was just with adolescents, by the way. And you can see one of them up there with her EEG net on. That's a trip in itself, as some of you here know. So the brain detection of that oddball, a deviant that was either a louder or a softer sound than the rest, all at the same frequency. In the middle bar, you see the segregated condition, where you see that same oddball, but now there's an overlay of additional tones. And then in the third, the integrated, the two different streams of sound are so close together you don't expect anyone to be able to detect the deviant. And we actually started out in this age of open science and full, we thought that the kids who were minimally verbal would have greater difficulty processing sounds neurally, and that we would see differences in the response to the oddball in any or all of these conditions. By the time we finished, there were no group differences. Early on, we did have some group differences, but never go with too small a sample. And here you can see that in the three conditions, the detection of the oddball is basically the same in all three, but there's enormous variability, okay? And then we looked at the extent to which the response, particularly in that segregated condition where they're trying to detect, the brain is trying to detect the oddball, but there's a competing stream of sounds which interferes with that. The amplitude of the P1N1 complex to that, looking to see whether response in that condition would be related to at least receptive language in this population. And actually we found, if you look at the lowest bar, that that was true just for the minimally verbal kids. So we did find a correlation, but only for the minimally verbal group. So it seems that the ability to detect sounds in a noisy scene is associated with better receptive language, because we know we're defining our group based on expressive language, and some have better or worse. And it seems that this may be traveling with overall auditory processing. Finally, let me get to some what we were able, or I would say not able, really to learn much about underlying neural mechanisms. Our goal had been to use imaging, functional MRI, DTI, to investigate this in the minimally verbal kids. And this was a collaboration with my colleague Frank Gunther, who has developed the most sophisticated computational model of speech production, which involves all kinds, it's a very complex network, speech production network. And in particular, he's been interested in this region here, which goes from the SMA, the initiation when you're about to produce you know what word you want to produce, to the speech sound map itself. So that's this link here in the left hemisphere. In an earlier study in verbally fluent kids, did find that there was impaired white matter. You can see it's not as robust in the left hemisphere for the verbal kids with autism, which he thinks may be related in that population to the differences in prosody and fluency that you see in some even verbally fluent kids with autism. And that's just looking at DTI. So then in collaboration with Dara Manoak at MGH, we try to do some fMRI work. You put the kid in the scanner, and they see a picture of something they know. We pre-test what words do you know, does the child know, and they see the picture and they produce the word. Well, this was a remarkable failure in terms of getting these kids. These were adolescents, despite quite a lot of training and repeated visits. But we did get data, some rather nice and intriguing data from one child. So we also did this in low-risk controls and in verbally fluent adolescents. These are all adolescents. And you can see that their activation maps look fairly similar. There's more variability in the maps from the ASD group. And here's what we found in the minimally verbal autistic participant from whom we were able to get decent enough data. So this is her activation patterns, and this is this robust connection between the speech initiation, and the speech output in the frontal region. And you can see that neither in the left nor the right hemisphere do we see any activation in these frontal regions. We see more abundant activation in the posterior parts of the brain. So this is intriguing, it's suggestive, it's an N of 1. I chide everyone who ever wants to go with an anecdote, but it certainly suggests that this is worth pursuing, which I'm hoping that piggybacking on Polo, since we didn't write this into Polo, we'll be able to do some ENIRS studies, which we think is a much more conducive methodology, and would, we can't get at the fine-grained details, but I think questioning whether we see differences in just frontal versus posterior, left versus right activation, ENIRS is perfectly suited to that. So, conclusions. Although language is not a core-defining symptom anymore, it may be, I think, the canary in the coal mine. In infants at risk, we see that behavioral and neural measures highlight differences with typical development, even for children whose later language is within normal limits, as well as in siblings, the siblings as well as those who end up with autism. For children who don't acquire spoken language, there are quite a number of unique behavioral and neural factors that are implicated. So nothing that I talked about from the minimally verbal is actually true for much younger children in terms of either behavioral predictors, speech motor deficits. We actually find that if you compare minimally verbal, mod 1 ADOS, to mod 2 ADOS in adolescence, we find that there's no signs of CAS at all in the low verbal. So that's quite a distinctive pattern. Future directions, well, ultimately, and that's why I collaborate with Connie and the rest of you here, we need to develop effective interventions that can be implemented early enough to promote spoken language in all children with autism. And here are my different collaborative teams. This is a photo of our current group at BU, and all the different people whose work I really had the privilege today to talk about. These are the people who have done all the work, and I just kind of stand on the sidelines, and have been fortunate to have funders, particularly the National Institute on Deafness and Communication Disorders. And I can't end without thanking all the kids and especially their families. I mean, you heard me talk about this stuff. Just imagine what it is like to go through a study where you come into the lab 7 different times, do home-based measures, et cetera, their time, their support, and their enthusiasm for the science that we are doing is just, I think, unparalleled. So, and thank you.